Well, first of all, thank you for the invitation to participate in this epic meeting. It's quite interesting for me to show what we do in Ocular Biophotonics, which technologies we use in this forum, which is more industry related, right? So as Antonio said, I'm a scientist at the Spanish National Research Council, and I work in the area of ocular biophotonics. In particular, I work at the Institute of Optics, which is one of the centers of the Spanish National Research Council, and we work there at the Visual Optics and Biophotonics Lab. We work around biophotonics to understand the optics of the eye, ophthalmology, in general, eye research. So when we think of vision, and I'm going to be super brief in letting you know what is vision, we automatically think on the eye, right? And there is this analogy between the eye and an image forming system, right? We have the images of the outer world, and they are projected on a CCD camera, which is the retina. Well, this process is not that visual. It's true that there is a part related as with a camera in which the images of the outer world are projected on the back part of the eye, the retina. But we don't have there a sharp image. We have something more like this due to the optical imperfections of the eye, and also due to the big changes that we have in the environment, changes in contrast, in luminance, in blur, in color, but also changes within the own system, right? Due to diseases, pathologies, surgery, or even corrections. So it's a bad quality image that is changing dynamically very fast. And in fact, it's the brain at the cortical stage, the one that, thanks to neural adaptation processes, we typically explain this as an instant Photoshop of the brain, the one that is responsible for us to see sharp independently of the conditions outside. So this complex process is not fully understood. And we, as you can imagine, use a variety of technologies. I'm going to show just very briefly 3 of the areas we are working right now. The first one is what happens when the camera, the eye glove, the optical system is too big. And this is what happens in myopia. The eye grows too much, and we don't have a focused image on the back of the eye. And you will say, okay, I am myopic. I know everybody who wear glasses for myopia. What's the problem? Okay, so myopia has a very high associated risk of leading to blindness. If you can see here very briefly, even low myopia leads to a significant increase in glaucoma, retinal detachment, or myopic macular degeneration. And this means that myopia is the first risk factor for blindness in the world. And the second fact that we need to remember is that by 2050, half of the world population will be myopic. So this is a really hot topic for the eye research community. And there are many areas of research associated with myopia because we really don't know the origin, whether it's genetic or what's triggering it. But we do know that the sclera plays a fundamental role. The sclera is the white part of the eye, and we are particularly interested in the back part of the eye. Okay, so because not only mechanics but also structure and molecular distribution changes. There are many technologies to understand what's happening at the biomechanical level, bibliography, airpath OCTs. We have been working with optical coherence elastography, which is the combination of an optical coherence tomograph plus an elastography technique, which basically means measuring the transmission of an acoustic wave to a certain tissue. And here you can see an example for a sclera of a porcine eye, and the difference is in the speed of propagation between a normal eye and an eye treated for myopia. And this is quite useful technology because it's non-invasive and it's already being used in humans, right? And this is a custom developed system, but I guess very soon they're going to be in the clinic. if mechanics change, structure also change. And there's a lot of people, there's been a lot of people working on structural imaging, second harmonic generation, mostly for collagen fibers. And the problem with the sclera is that it's a highly heterogeneous tissue, very anisotropic, and it's opaque. So it's complicated to do a 3D imaging stack as we want. So now we are combining two photon excited fluorescence microscopy with second harmonic generation to try to link molecular changes to structural changes. And we started doing that at Harvard with Fred Cho and Jun Ha-Huan using an Olympus fluoview system and also an Evintech OCT microscope, sorry. And we got very nice ideas for custom developments that we are doing around right now. So it was really a very good use of two complementary systems. So that's regarding the ocular optics. What happens if the camera is kind of broken? But what happens with the brain? The processor, how is the processor working? And for that, we use adaptive optics, and Antonio already mentioned that. And I guess most of the people in this forum are aware of what is adaptive optics. But I would like to remind you that adaptive optics is a technology that we have inherited from astronomy, and that only in the last 25 years has been used for visual optics, and that allows us to basically control the wavefront at a certain conjugate plane of the eye, the pupil plane, and therefore we can not only characterize the ocular optics, the higher the aberrations and so, but it also allows us to manipulate. So what application has this in vision? I'm gonna just show a couple of them. So for example, we use adaptive optics-based systems to simulate very complex designs, in this particular case with an SLM, with a liquid crystal. And we can either manufacture them in terms of faceplates, or we can just simulate them on bench. And you can see here, this is a commercial design available in the market. And these are crazy designs we decided to test. And we were able to test them. We can test them using different technologies. And I will talk more about that later. And this is quite interesting for certain companies because it allows us to test any design they want to create or they want to update or test for certain patients before they're even manufactured or implanted or tested in patients. So this was really a good starting point for collaboration with the industry. An example of how this works with an industrial collaborator, for example, is how we were able to map the face map of a commercial design, the M+ Oculentis, bifocal design. You can see here the two peaks, and this is the image of the real intraocular lens that is implanted in the patients. And in this particular case, we tested the through focus optical quality and visual quality on bench, and using 3 different technologies, but we also were able to test them on patients. It was the first time we validated the different technologies, simulating a real multifocal design. As you can see here, the curves are very similar, although there is more noise in the subject responses due to the higher aberrations and other things. So, which optical technologies, and I'm emphasizing this here because I think some of the manufacturers of these technologies are here. So some of the ones I've been using in the last years are static ones, for example, just use a faceplate or an intraocular lens next to bed, and you just look through it, through the system. There's also the dynamic faceplates, and I think this is an image from FaceForm, which have experienced a really good development in the last year. And the classical elements, which are the deformable mirror and the SLM, the DM is really a fast optical technology, and it doesn't have any chromatic effect, but it's limited in the phase it can acquire. On the other hand, spatial light modulator is a matrix of pixels, so you can achieve any phase differences you want. For example, we wrap it up to 2 pi, but it can be used up to 4 pi, but it's slower and it has chromatic effects, particularly when you want to test designs in polychromatic conditions, white light versus monochromatic. And in the lab, we developed another simulating technology. We started using an optotunable lens from optotuning to modify the waveform temporally instead of spatially, which is what all these other technologies do. And we call it SIMBIS technology, and it's based on the temporal multiplexing of an optotunable lens, which is now commercialized by our company, Twice Vision, and which aims at be like a low-cost alternative to this more classical, let's say, technology. So how do we use these technologies in the lab? And let's see, this works. Okay, so we set up these crazy adapted optics-based visual simulators. This is an example of the one that I developed at the Institute of Optics. We have a supercontinuum laser source that, thanks to a dual acoustic tunable filters module, allows us to have 2 channels, a near infrared and visible one, and it allows us to have very narrow wavelengths in a very fast way. We can select them very fast according to our needs. This will be the subject, and we have an AO loop. In this particular case, it's from Imagine Eyes. We use a deformable mirror to compensate for the aberrations of the subject. But we also have a special light modulator to generate very complex designs. And also we have the testing channel here, a conjugate PP plane channel, where we can either place intraocular lenses next to it, we can use SIMBIS technology, or we can test whatever faceplate or optical design we want. We have a retinal camera here, which is kind of an 1.5 imaging, 1.5 system pass, and we can have kind of a PSF, the point spread function of the subject, plus the different simulations, so we can analyze the optics of the eye. And finally, we have a psychophysical channel. Here we have a DMD, a digital mathematical device, which we illuminate either with white light or light coming from the supercontinuum, and we project it on the retina of the subject through the simulations we have prepared. Okay? So as you can see, it's quite a big system. This is an image of the first stages of the development. Now it has 3 levels, I think. And our goal was to go from these big systems to clinical devices. And this is so far at the moment the final clinical visual simulator, which is not an adapted optics based simulator, but it's an active optical based simulator. An example that I like a lot to prove the use of these active optical technologies is presbyopia. Presbyopia is the loss of the capability of accommodation of the eye. We cannot see near once we are older than 45, 50 years old, and it affects almost everybody in the world. And the thing is that not only we lose the capability of seeing near objects, but in many cases the lens of the eye is yellow, and there is a cataract there, so it needs to be changed by an artificial IOL. The designs and the differences in the types of IOLs are almost infinite. So in a collaboration with CTO, with Christophe Pagnol, and we test their fine vision diffractive design. It's this one. It has 3 peaks, far, intermediate, and vision, and it combines a refractive and a diffractive design. So it's a complex idea, and we test it on the big lab simulator, but also on the clinical visual simulator. And what we did was show what we were simulating before the surgery and test vision after the surgery when the subject already had the intraocular lens implanted. And the results were fantastic. It demonstrated that we could simulate preoperatively the vision of a patient after the operation. Look at the 2 focus curves are very similar. And in the cataract patients, we are able to replicate the behavior after the surgery, although there are differences due to those opacity. This fantastic capability has helped us to collaborate with a lot of industrial partners with different designs of intraocular lenses, and we are proud to say that we have helped in the development of some of those designs. The last example I wanna show you here is related not only of understanding how the visual system is, but also to understand what happens in the myopization process, which is how the eye grows. If that fails, we have, for example, here a myopic eye, or we have a keratoconus eye. You see the cornea is granular, right? So we know that visual information is quite important. And what happens is that the retina detects certain information, and it starts a series of molecular chain reactions, which we call the retinal signaling cascade, which at the end makes the sclera changes, the scleral remodeling, and that's why the back part of the eye is longer than it should be. So as you can imagine, this is a complicated thing to study, and this is a fantastic project we are developing with Professor Michael Celiac from the University of Rochester, where we are combining a commercial scientific multiphoton microscope with an NEA recording system, and we are incorporating a visual stimulation channel with the DMD and a synchronizable light source. And we are stimulating different ways the retinal ganglion cells to understand how they work and how we can implement those tests in humans in the visual system. So this is an example of a combined commercial devices with some tunable changes in the lab. So in summary, and I think I'm okay on time, biophotonics for eye research means for us multidisciplinary, because we really need to understand and to use different imaging technologies. And what we would like to have for the projects we have right now is we are particularly interested in novel imaging tools that allows us to image scattered tissues. And we think here the focus is on the for example, phase retrieval approach is a computational approach more than optical one. We really need laser sources for multiphoton microscopy that allows in vivo imaging. I think Carol, who is talking later, will be talking about one of their developments architecture that goes in this way. So we really need more work with this. We really need to be able to acquire modular microscopes when we want to have complex spectral setup, because sometimes what we really want is not a big old-fashioned system, but we want to combine different strategies and add our own custom design. We are now working as well on nanoimaging strategies in combination with multiphoton high fluorescence microscopy, and all that only for the myopia project. Regarding the AO-based, the adaptive optics-based projects, we are very focused on polychromatic optics design. We want to help designers to design complex optics for IOLs or contact lenses that can be tested in white light conditions. And something that will help us a lot would be to have customized super continuum laser source, customized and cheap as well, because we really need something very specific, but not very powerful at the same time. And of course, we are open to collaborate with clinical and industrial partners, which is really what we have been doing always, right? Otherwise, we wouldn't move forward. And that being said, I would like to acknowledge all collaborators, which has been responsible for most of these developments, like the people at Susana Marcos Group or Andy June's group at Wellman Center, also the University of Rochester, in other centers, and of course, parent team. I have in Madrid is the adaptive optics and the multiphoton team working on myopia and imaging projects. Also collaborators within the research group, particularly with Victor Rodriguez, we are working a lot in visual perception cues for myopia, which is quite an interesting area of work, and with James Sherman in all the multiphoton technologies development. And finally, of course, a network of international collaborators is essential to this kind of work, particularly one of these collaborators with which we are working very closely right now. And I guess Karen will be talking later about Kasia's work on two-photon vision projection. So that's it. Thank you very much. These are my means of my media for contact or my personal email. And if you like outreach, please take a look at this free book that we published with SPIE, and it's very funny for kids and for teenagers. And that's it. I'm ready for your questions. Thank you.